Bismillahirrahmanirrahim. Dear students, welcome to my new lecture. So in this lecture we will discuss about the Cytochrome P450 system. So what is Cytochrome P450 system? With the basic introductions. The Cytochrome P450 system as a superfamily of monooxygenases. As we know that there are particular enzymes which are present in our body and they are responsible to perform different activities according to their wills. As we know that there are certain enzymes which are present in our digestive tracts, which are responsible to digest protein, carbohydrates and many more nutrients which are present in our body. So this is another group of, you can say, enzymes, the Cytochrome P450 system. This is the superfamily of monooxygenase. Heme containing enzymes are hemoproteins, officially abbreviated as a COIP. So it is a large and diverse group of enzymes that catalyze the oxidation of the certain organic substances. The Cytochrome P450 system has the capability and tendency to absorb the light at a wavelength of 450 nanometers. So it can absorb the light at a wavelength of 450 nanometers. This is the wavelength of Cytochrome P450 system, and this is called absorbance. So look at the graphical representation. This is starting point and this is the peak. You can say at 450 nanometer, the Cytochrome P450 system has the tendency to absorb the light. Now come to the nomenclature of the Cytochrome P450 system. What is Cytochrome P450 system? CYP. So 3 is the family, so it indicates the family which is greater than 40% identity, while A, capital A. So this is subfamily and this is greater than 59% identity, and 4 is the specific isoform. You can say genes. Iso means same and form means shape, structure. So for example, when two protein molecules, they have similar shape. Look at here, this is CYP, root symbol for Cytochrome P450, and 2 is the family, and C is the subfamily, and 9 is the gene identifier. After that, as we know that the substrate of Cytochrome P450 system enzyme include, the first one is lipid and steroidal hormone, the second one is xenobiotics such as drugs and toxic chemicals. Substrate is anything which can anything which can attack on enzymes. Substrate is anything by which the enzyme can act. So that is called substrate. As we know that the cytochrome, these are the major enzymes involved in drug metabolism and bioactivation, accounting for about 75% of the total number of different metabolic reactions. Now what is the site where these cytochrome they are present? Especially these cytochrome, they are present throughout the body, they are distributed throughout the body, but the cytochrome, they act primarily in the endoplasmic reticulum of hepatocytes, liver cells and the cells of the intestine. So these are the major sites and which these cytochromes are present. As we know that they are distributed throughout the body, but they primarily in the endoplasmic reticulum. So as we know that this is an important component of the cells and the cells of the intestine, so they are present here. Condition that causes viral hepatitis or cirrhosis will affect the efficiency of drug metabolisms by the cytochrome P enzymes. P450 in humans. So as we know that human cytochrome P450 system, they are primarily membrane associated protein located either in the inner membrane of mitochondria or in the endoplasmic reticulum of cells. So some cytochrome metabolize only one or a few substrates. So they can metabolize only one or few substrates by which the enzyme can act, such as cytochrome 19 and aromatase, while the other may metabolize the substrate, the multiple substrate. Humans have 57 genes and more than 59 pseudogenes divided among 18 families of Cytochrome P450 genes and 44 subfamilies. Look at here, humans have 18 families of Cytochrome P450 genes and 44 subfamilies. And Cytochrome P1, the drug metabolisms, they are responsible for drug metabolisms So there are two subfamilies, three genes, one pseudogenes, and cytochrome P2 drug steroids, vitamin D metabolism. So they are responsible for vitamin D metabolism, and drug steroid contain 13 subfamilies, 16 genes, and 16 pseudogenes. And cytochrome P3 drug, they are also responsible for drug metabolism, one subfamily, 4 genes, and 2 pseudogenes. And cytochrome P4 arachidonic acid or fatty acid metabolism, 6 subfamilies, 12 genes, and 10 pseudogenes. So if we compare cytochrome P1 and P2, so you will see a lot of difference. So here in cytochrome P1 there are 2 subfamilies, while in cytochrome P2 there are 13 subfamilies. While in cytochrome P1 there are 3 genes, here in cytochrome P2 there are 16 genes. And cytochrome P1 there is only 1 pseudogene, and cytochrome P2 there is 16 pseudogenes. Now, how drug metabolism is take place through cytochrome? So cytochrome P450 system enzyme accounts for 75% of the drug metabolism. It means that 75% of the drug metabolism is take place through this pathway. Most drugs undergo the deactivation by cytochrome either directly or by facilitated excretion from the body. Also, many substances, they are bioactivated by cytochrome P to form their active compounds. Drug interaction, come toward drug interaction. So many drugs may increase or decrease the activity of various cytochrome P isozymes, inducing the biosynthesis of isoenzyme, enzyme induction, and directly inhibiting the activity of cytochrome enzyme inhibition. There is a major sound of adverse drug interactions. Drug interaction, inshallah, it will be discussed in my next lecture.